Thanks, Kutelango. And vanakkam to all of you. Welcome. Welcome, Dr. Raj Gopal, for taking time and, coming and willing to present, share your knowledge, right? So Dr. Rajagopal Appavu is an award-winning assistant professor at the Taneja College of Pharmacy, University of South Florida, Tampa. He has completed his PhD at the Indian Institute of Science, Bangalore, with mentorship of Professor P. Balram, who is a former director, Indian Institute of Science, Bangalore. And also he was a science advisory committee to the Union Cabinet and Scientific Advisory Council to the Prime Minister of India. Dr. Raj came to University of Medical Branch, Galveston, for his postdoctoral work, where he developed nanotechnology-based vaccines for cocaine addiction. And the research work or article published in prestigious journal, American Chemical Society Neuroscience. Later, he moved to MD Anderson Cancer Center, Houston. That was like a world #1 cancer hospital in US News, where he targeted drug and vaccine delivery work. Now he is assistant professor at the Taneja College of Pharmacy, University of South Florida. Currently, he is doing both teaching and research.
I cannot hear anything.
Can we mute him? Thank you. Where he is currently doing both teaching and research in the area of nanotechnology-based personalized medicine and therapeutic medicine to healthcare students who are like, MD and PhD kind of students in infectious diseases and non-infectious diseases. So this webinar, we targeted like two major agenda. One is to kind of demystify myths about the COVID vaccine. Dr. Rajagopal is going to, explain or demystify from the science behind the vaccine. He's going to talk about, the vaccine development, how they're different, like, various types of vaccine. A lot of myths he's going to, demystify. Later, after that, we're going to talk about the biological career options, which will be helpful for a lot of high school students and parents. That's, the second of our like last, I would say like 15-20 minutes part of that. With that, I welcome Dr. Raj Gopal. Please take the stage and, share your knowledge with us. Thank you.
Hi, thank you so much, Lokesh, for your kind introduction. It's a very honor to be here, and it's my prestigious and a privilege to present some of my work. And then I have been working in the vaccine past 8 years since coronavirus, and then cocaine, like different kind of vaccines. And I have been reviewing a lot of documentary regulatory stuff, so what molecule inside the vaccine, stuff like that. So let me share my files, and then from there we can share some of my work so that it will be useful some of the participants over here. So let me share my file, PPT, which I have here. Give me a minute. Thank you so much for giving me the opportunity to present here. So my name is Raj Gopal Appavu. I'm the assistant professor at the Taneja College of Pharmacy. I have been teaching and doing the research, both vaccine development, infectious diseases and non-infectious diseases. I have students, around 42 students, both they're doing MD and PhD. And I have been trained a lot of students there in big physician, some of the schools, medical schools, like Duke, some of the Stanford schools. So based on the knowledge, we have talking with a lot of scholars and a lot of document reviews about that Pfizer, BNT, and then Johnson Johnson, AstraZeneca. So like we are working with so many people's because we want to get the vaccine fast. For that, we get work with a lot of molecules And I'm going to share some of the knowledge, some of the information which I got it over period of time in the infectious diseases and non-infectious disease. So objective of the presentation is, we know that this is the magic molecule, the magic liquid, little bit that we are going to inject like a few microliter in our body. So what inside the vaccine? Okay, what are the ingredients exactly present there, and how this mRNA vaccine works? So all these things I'm going to share with you. Hope it will be much benefit. So this is I'm going to talk a lot of molecular levels. I hope it will be a great benefit. So so this is a little bit about some 10 objective I took, and we'll focus as we go along. So next slide, please. So this is the typical vaccine. Okay, this vaccine, you can see that something very, very liquid. There is a lot of molecule inside. Okay, next slide, please. Yeah, so the what this vaccine made up of? Okay, this vaccine made up of basically periodic table. Why I want to show the periodic table? The periodic table is something really, really important. In the periodic table only everything merged. This universe, air, water, land, fire, etc., etc. This universe is made all these periodic table molecules. If you take water molecules, two hydrogen, one oxygen. Okay, same thing. If you take DNA molecules, our nucleic acids, what are the molecules? Basically, the carbon, nitrogen, oxygen, hydrogen. Okay, same way you take it the vaccine also you can see the carbon, hydrogen, nitrogen. Okay, not big molecules, but it's arranged a different way, different form. It gives a different functions. That is what. So basically, the components are same, but the ingredients, the molecular structure, the molecular function, it is way different. That cause infectious diseases, and it gives a vaccine also. So it does both. Okay, these molecules gives both. So the vaccine that we are talking with the mRNA vaccines. In the mRNA vaccine also, basically carbon, nitrogen, oxygens. Okay, the COVID, the infection which cause the diseases, that also carbon, nitrogen, hydrogens. So how it varies, all these things, we are going to see a couple of things. Yeah, next slide, please. Okay, so in this vaccine, okay, there are two main components when they make the vaccines. Because in the laboratory, when I make a lot of vaccines before it goes to patients, to the animal models, I put typically two things. One is the active ingredients, another is the non-active, inactive ingredients or non-active ingredients. So in the mRNA vaccines, the active is molecules, that is RNA molecules, mRNA molecules. The rest all things and all, to stabilize that mRNA, we have to add some of the things. Which are the things? Like PEG. So that is the polyethylene glycol. So polyethylene glycol, what it does it, once you inject this mRNA molecules in our body, it should circulate entire body. So that is the reason we put the PEG molecules. So PEG molecules, what the function of the PEG molecules, you take this like this vaccine, like a few microliter vaccines, in that minimum 5 to 6 ingredients will be there. So one of the main ingredient is the mRNA, rest of them are PEG, and another is the phosphocholine, another one is the cholesterol, another one is the hexadecanate, and another one is the sodium chloride. So those things I'm going to talk a little bit. So the main ingredient is nucleic mRNA. The second one is the PEG. This is the PEG. You can see that the inactive ingredients, the first molecule, that is the PEG. So PEG function is circulate your vaccine to enter your body without any issues. So that is the function of the uh, PEG molecules. Another one is the, you can see the phosphocholine. The phosphocholine, what it is, function of the phosphocholine is, when you take the molecules, you have, it should be stayed sometime in your body, at least a couple of days. Okay, for that you need to have some emulsifier. So that emulsifier function, what it does is, it stays in your bloodstream and activate your T cells, B cells. Okay, and it passes through your entire organs, like spleen, heart, pancreas, like entire body systems. So that is the function of the emulsifier. The cholesterol, where it is a function is because our body is mostly it's cholesterol. Okay, so cholesterol something that we need to bind in our body systems. Okay, that is the what we have to add the cholesterol. If you see that mRNA encapsulated mRNA with surrounding layers all are cholesterols. And let us come to the sodium chloride, monobasic potassium phosphate, potassium chloride, and the dibasic sodium phosphate dihydrate and the sucrose. So what is this function is? It is a basically it's buffer. It's a buffer means our body is a neutral physiological buffer like 7.2, 7.4. Such a pH should be there. Same, the cocaine vaccine also we have to have the physiological buffer, something 7.1, 7.2. We should not bring the low pH as well as the high pH, because if it goes high pH or low pH, there will be a lot of cytotoxic effect for your organs. That is the reason what we do is we put the sodium chloride and the potassium chloride and the sucrose, stuff like that. Okay, those are the molecules which stabilize your vaccine to stay at least a couple of days in your body. So, but why this is the problem it comes next. The sodium chloride and the potassium chloride. The potassium chloride, what it is, that some people get the heart cardiac arrest. The reason is the the people, for example, some patient has the tachycardia or palpitation. Okay, those people maybe have some issues taking some of the vaccines with the potassium chloride. So I'm going to talk a little bit about in the next slide. Next slide, please. Yes. So this year I'm going to compare all these vaccines. We have the Pfizer BioNTech vaccines, that is the mRNA vaccines, and this is the Moderna vaccines. If you come to that inactive ingredients, you can see that active ingredients, both molecule you have it like mRNA, both in Pfizer and the Moderna, but in Johnson it's vector RNA molecules. So that is something different. So let us focus the Pfizer and Moderna. So here the active ingredients both are same, and then inactive ingredients, the PEG molecule, that is the PEG PEG molecule. You can see that the both are same, and pretty much the phosphocholine also same because that is the emulsifier in the both vaccines. In the cholesterol also you can see that same. But when you come to the next molecules, the next component or the next ingredients, it will be way different. Okay, the reason is here sodium chloride. Okay, so what Moderna thought of doing something different, the Pfizer, because of some of the people who has the cardiac irregularities and the palpitation, tachycardia, those patient, this vaccine doesn't shoot. So what they did was is they replaced the sodium chloride and they put tromethamine, and they replaced the potassium chloride and they put the acetic acid, and they replaced dibasic sodium phosphate dihydrate. Then they just simply put the sodium acetated, but they kept the sucrose. It is common because your mRNA molecule want to stay, and it need some kind of lifetime to stay in our body. That's what they put this sucrose. So this is the reason why some people after taking the vaccines, they have kind of heart failures, things. This is the things, few reasons they want to change it in the ingredients. I hope now you got the difference why some vaccines give some kind of side effects like heart failure, stuff like that, and some vaccine doesn't give like such a scenario. So it is based on that person to person, and it gives kind of idea that which vaccine should take, which vaccine not to take. It's based on the patient's medical history. For example, for some person who has the continuous heart issues, myocardial infarction, stuff like that, they should see that what ingredients, what vaccine they should take before to go to the site to take the vaccines or the clinical facility. So these are the things, something we should know that which vaccine we should take, what ingredients exactly is there. So these vaccines, um, I didn't put one important molecules here. That is the water. Okay, that is the sterile water. So all these molecules should dissoluble in the sterile water. Okay, they don't put normal water. The percentage will be little bit ups and down for each vaccines because it should dissolve in our blood stream. Then it take all these peg molecules, sucrose, cholesterol, phosphocholeins, everything our body system. That is the reason they took some amount of the sterile molecules. So pretty much it's water molecules. It added so many ingredients, so many formulations, stuff like that. Then we take these vaccines. So next slide, please. Okay, how this mRNA vaccine works. So basically, what we are doing here is we are taking, we know that what are the molecules, what are the sequence in the mRNA. We have the mRNA spike molecules. We are taking a portion of COVID RNA molecules and injecting our body system. And we are training our body to know that there is a foreign pathogens enter our body and then start going our body system like lymphocytes, all this important organs, and start producing the antibodies. So once antibodies, if you teach the antibodies that who's that person is coming from outside, start making their own antibodies and start killing the whatever the foreign molecules, COVID or MERS-CoV, whatever comes, it start binding and start neutralizing the effect of that particular virus. So basically the mRNA, it is something really nice technology because this COVID is just like a 2000 it came, but the same molecule like 10 years, 20 years ago, there is a MERS-CoV. The MERS-CoV and the SARS-CoV, the structures, the sequence pretty much same. That is the reason we could able to achieve this COVID vaccine quite faster because we have the back lot of research, a lot of paper has been published about that MERS-CoV. So we took all the information from the MERS-CoV virus, and then we compared the SARS-CoV-2, and then we able to bring this molecule into the public very soon. The next slide, please. Okay, so now what different kind of vaccines? So there are a lot of vaccine is going on in the field. For example, when you go to the child, to the pediatrician, they see that have you taken influenza, MMR, mumps, VAR, like DTF, like so many vaccines. So these vaccines are different kind of vaccine, as you know that. So in vaccine, there is a lot of classification is there. That is the whole inactivated vaccines, which means you take any virus that make them to inactivate it. Okay, so in the laboratory, if you go to the biocontaminated laboratory, there is like Wuhan in China, you might be heard that. So they have like a different kind of virus in the biocontaminated laboratory. They do work with, they activate um, dead virus to good virus and good virus to bad virus, so like that. So some of the vaccines, the inactivated vaccine means that is the real virus, okay, that we are able to inactivate in the biocontaminated laboratory to make not to give the infection, but it gives the protection. Okay, that is called the inactivated vaccines. So live attenuated vaccine means you take the live um, virus or live bacteria and keep them and pack them in the material which we are able to inject in our body system and gives the antibodies or immune response. So that is called live attenuated vaccines. So subunit vaccine means you have the virus, the entire virus you don't need to take. Okay, take a part of the virus and do the bioengineering and make it and purpose it for the vaccine production, anti-production. So that is the subunit. So basically subunity vaccine is means taking portion of the virus or bacteria and making them to produce the antibodies in our body system. That is called subunit vaccine. So synthetic carrier vaccine means we know that naturally there are lot of vaccines. So synthetic means you laboratorily in your laboratory you take that vaccine and synthesize artificially and then immunizing into the particular patient or mice or monkey or whatever the animals we want to test. So that is called synthetic carrier. So synthetic carriers means we know that what is like COVID vaccine is we have the COVID virus. So that we are practically generating in the laboratory. So that is called synthetic vaccines. So when you how you can do it, that synthetic vaccine is for that you have to work with a biocontaminant laboratory where your water should be purified, your everything basically it is really dangerous. That is the BSL-4 biosafety level 4 laboratory. Some of the people work making the synthetic vaccines. So basically you are directly working with virus that you are going to make like thousands of molecules, big crown stuff like that. Everything you are going to make it in the laboratory setting. That is called a synthetic vaccine. So viral vector means you are going to, it is kind of synthetic vector, synthetic vaccines that you are going to take only the particular vectors which cause effective target to the particular disease. So another one is the electrophoration. So electrophoration means that you take your molecule or the vaccine and put inside and give the electric pulse so that the molecules, the vaccine, it will be distributed into your body systems and give immune response. That is, but pretty much this is the technology which is not advanced so far, the electrophoration. So pretty much what right now people are using is the subunit vaccines and the mRNA vaccines. That is the current technology. The next slide, please. Okay, this is the something. So the vaccine is that safe, okay, or not safe. That is the things which is going on for a year. So my perspective and my animal testing, my clinical trial experience about that a vaccine is safe or not, I would say that it is safe. Definitely not safe. Some of the people who has the medical complications and immunosuppression and immunocompromised patient, it definitely it's not safe because already they have a lot of medical history, medical issues. So definitely this vaccine susceptible for those medical complication patients. So the another thing is, so we are usually the molecules, the mRNA produce the DNA and the protein stuff like that. So when it comes to the cytoplasms, that mRNA only gives the antibodies. It doesn't make any new cloning of the DNA. So there is no evidence that that whatever the mRNA molecules, mRNA vaccines make new modifications in the genes. So definitely it doesn't do it based on the research, previous research and findings and the discoveries. So because the reason why the people are suspicious about this vaccine is not safe and because it produced so quick. Okay, it doesn't mean that because they produced so quick it is not safe, because it produced, because we know that this is like 10 years ago and this COVID has been started like 50 years ago. So we have the all these molecules in the biocontaminant laboratory like 40 years ago, and the people are working and the people are learning about this vaccines, virus, everything. So we have know about these vaccines, these virus molecules like 40 years ago. And so it is not new. Definitely it's not started like 2 years ago. It started while ago, like a few decades ago, and people are doing the research. That is the reason we could able to get the quick vaccines. So the next slide is, can you go please? Okay, so this vaccine, does it give protections? Yes, it gives both protection as well as the pain. So you can see that so many patient who are people who are the public people who are took that vaccine, it has like pain, fatigue, headache, muscle pain, fever, like so many complication has been reported. So it gives the pain as well as the protections. You can see that the right hand side, a lot of data which you can see based on the um, flow cytometry, antibody responses, ELISpot, ELISA, everything it given and peer reviewed that this is definitely give the protection, and then and is much safer to take. And if you see that the middle of that column, there is the you take the day one the vaccines, and day 21 and day 28 you see that lot of antibody titers. That is that definitely makes lot of antibodies and neutralize whatever the COVID variants, kappa, delta, like different variants are there. So it pretty much does very good job, those vaccines. So definitely it is safe, and based on our animal experiment, our reviewed safety data sheet, and the, so many pharmacologic effect, everything we evaluated, and then it is showing that it is under same under control, and there is no much worry about these vaccines. Yeah, next slide please. Okay, so what kind of protection it gives? Okay, and where it gives all these things, you saw that when you take the vaccine molecules, the main thing is two cells, that is the memory cells. Okay, the memory cells, it circulate under your body system and give the lifetime memories. Okay, so why I said the memory cells is when you take these vaccines, it internalize in your organ, and your memory cells understand that there is a COVID virus coming, and start making the lot of memory cells. So memory cells gives antibodies against lot of viruses. So that is how I put these memory cells. So this vaccine goes entire your bodies, like brain, spleens, bone marrow, kidney, and the rest of the bodies, and activated all the memory cells. The memory cells make sure you have the right protections. Um, that is the purpose of this slide. So as long as you have the memory cells, there is no worries. Why some geriatric patient get side effects from this COVID vaccine is they do not have much memory cells. Once you don't have the memory cells, okay, your body doesn't understand that, okay, there is this is the bad guy or good guy, doesn't have any idea, and start giving a lot of side effects like uh, heart failures, and there is a frozen blood, stuff like that. So these are the some side effects because of the loss of memory cells. So as long as if you have enough memory cells, you are good to take a lot of vaccines, like 1 year, after 2 years, like that. So it gives you a long protection. That's all some of the literature published. Yeah, next slide please. Okay, are the vaccine given in India inferior the given in the US? Okay, so what is the difference in US and India means? The vaccine both countries are same. The mRNA molecules in your vaccine both are same. The ingredients bit differ, but the problem which we have in India is lot of variants. Okay, lot of kappa, delta, like so many variants existing day to day. That is the reason some of the vaccines um, have some potential to cure these kind of infectious diseases. So basically, what I want to say is the vaccine both are same, everything does same job, but the issue is in India is a lot of variants that gives a lot of issues. though we are in the first lockdown, we have a lot of good response and a good control from the government. But the when it exists with a lot of variants, COVID variants, it is we need really some good technology, that what are the variants coming every day, and what are the modification we need to do in the vaccines. So we need some more technology in order to address this kind of issues. So the vaccines are same everywhere, but a lot of variants populating nowadays in the India. That is the issues. So each variants, we can't get the different kind of vaccines, and we don't know which variants um, the person who has, or the patient who has in our body, or the droplets. So that is something really, really hard to find in order to pinpoint to give the right vaccines. So whatever the vaccine which we have is right now is a common vaccine. Okay, the vaccine work for all the COVID variants, but we don't have the vaccines something for each variant, different kind of vaccine. So we need to advance some of the research based on the variants. Next slide, please. Okay, so science behind the vaccine development. So definitely science and the technology, they played an important role to bring this vaccine so fast. For example, if you take some of the polio vaccine, if you in the period of time it took like 100 years, 200 years, um, and then they could able to eradicate the polio vaccine after so many years. But this COVID vaccine, we have been successful within decade. So the science behind is lot of technology, lot of bioinformatics tools, lot of artificial intelligence. So those are the technology. It helped bring the vaccine so fast. Without this technology, it is very, very hard. If you look at that, like 100 years ago, they don't have much understanding about the molecules, and they don't have much facilities, whatever the softwares, the technology which we have, the nanotechnology, all these things. But now we have all this nanotechnology, bioinformatics, artificial intelligence. Those are the things which help to bring the vaccines very fast. Though still we have the lot of technology, but we don't have the right drug candidate. That is, we are still lacking on it. So hopefully it will be done another couple of months to a year in order to solve or bring the best drug into the market. So the basically science is behind this vaccine is a lot of research. So a lot of research were there. So many people are doing. When the MERS COVID populated in the Middle East syndrome diseases, from there people started working on this. What are the variants? How this variant can go different way. So people have a lot of great understanding, the research peoples about these vaccines, and they have the pretty much very good data. So the science definitely play a role. uh, Go ahead, please. Next slide. Next slide, please. Okay. um, so what I want to say in this slide is the traditional vaccine, it takes 100 years and more, but the current vaccine technology to help to minimize the time. On the right side, you can see that the worst, the pandemic is the Black Death. So that is wiped out off of the populations in the Europe in the year of 13,000. Okay, 13,000. You can see here, 13,000. So when compared to that Black Death pandemic or COVID is, it's not that much danger. That's what I'm trying to say is. So the history, the world saw a lot of big pandemic, and there is a 50% of the world death happens because of the Black Death, and the smallpox, and the plague, and the flu, and HIV. So after that, this the COVID. So the COVID is the pandemic definitely, but it's not such a dangerous when compared to the previous pandemics, because that time we don't have technology, and we don't have any understanding about molecules, and we don't have this kind of facility. But now everything we do have, that is the reason we could able to achieve and shorten the times, and we can cut the corners, bring any vaccines, any drug very fast without any hurdles. Yeah, next slide, please. Okay, this is something really I want to tell that everything matters. Here we should understanding about our molecules, our body system. Typically, you can see that our blood cells, red blood cells, it's about a 700 to 800 nanometers. And the COVID, it's 100 nanometer size. This is the problem, okay? The COVID is like 100 nanometers, such a tiny molecules, okay? Whereas our red blood cells, it's 700 nanometers. And then our white blood cells is like 2000 nanometers, okay? This is the reason, main reasons, lot of death happens because we are inhaling, basically we are inhaling the 100 nanometer COVID molecules. Okay, that is goes in our body through nose, through mouth, ear, stuff like that. It goes and mix with red blood cells. Okay, if our body, our blood cells are healthy and there is no issue if you don't have any sickle cell anemia, stuff like that, it able to kill the coronavirus in our red blood cells itself. In our body, the red blood cells shape changes. If we have some issues in our white blood cells or red blood cells, what this COVID molecule does is it masks that entire red blood cells. Okay, one of my students, he's in the New York Medical College. He was telling that Dr. Raj, I saw the one of the patient. I saw his lungs, whole blood is clotted. Okay, the reason is these COVID molecules goes to the contaminate into the red blood cells and stop arresting our oxygens. So the red blood cells purpose is carry the oxygen into our body systems. But when it goes to the COVID, to the red blood cells, it can't able to transport any oxygen in our body systems. Then ideally what will happen is that red blood cells start freezing and changing structures, and then there is no more transportation of the any nutrients, any hormones, or any um, RNA or hormones, whatever. Okay, so that is the reason. Okay, the COVID, the virus is it's contaminate the entire world. We are inhaling, we are exhaling. Okay, as long as our red blood cells, our white blood cells are healthy, we can able to fight with COVID. But something wrong in the size wise and the red bloods sickle cells size or change, something happens that it gives a lot of issues based on the some other physician scientist experience. Yeah, go ahead, next slide. So this is the typical comparison, the battlefield between the COVID respiratory droplets. Respiratory droplets means the water what we are inhaling, like sneezing, all this air fluid, and then red blood cells, everything you can see here. So this is the basically when your body COVID basic firefighter. I would say these are the people's which is fight it, and who wins is the matter. The red blood cells wins, we are survive. Okay, the COVID wins, definitely it harms to the rest of the red bloods and white blood cells. So we have to, so purpose of this slide is we have to understand of the molecules of the size. Okay, the COVID, what is the size? What is our red blood cells? What is our white blood size? So this is gives kind of understanding that how we are fighting with small molecules. So the small molecules does big danger to the human body. That is what I want to emphasize in this slide. So go for next slide, please. Okay, so when it become normal, Because like a couple of years ago, we have a lot of public gathering, we go to friend house, stuff like that. But past a year, we could not do anything. So the reason is, um, if you see that right side, the people demographically vaccinated. So right now, the highest number, the Israel, 60% of population in the country, they have vaccinated, and followed by UK, and USA. So like that, so all the developed countries pretty much they vaccinated, and their immunities are pretty good. And then um, these vaccines pretty much fight any COVID variants. So once like all people, yes, more than 70% is vaccinated, each country, so pretty much it is back to normal. The another danger is the day to day, the COVID variants are increasing, and we are getting vaccination means we could be able to fight with new coming COVID variants. But if you don't get vaccination, and definitely it is hard to become your normal and the rotation life. So that's what the vaccination something really important to fight all these COVID variants and the COVID diseases. So eventually, the now our body systems train to fight all these variants. So naturally, we are getting immunity in our body. So definitely, uh, probably another couple of quarters, we are back to normal as per as the statistical report which were published some of the research journals. But if you don't take vaccines, it is really hard for the individuals and as well as the society also. So it is all about that person's medical complications, pretty much, I would say. So taking vaccine is definitely doesn't harm anything. Yeah, next slide, please. Okay. Uh, this is so far I have talked about that how we make the vaccines in the laboratory setting, and how we can able to take into the animals. In animals there is so many variants, so many things are there, like different facilities there, how to handle this in different environment, these vaccine molecules, and then how it bring into the clinical trials. Clinical trials is pretty easy compared to the vaccines which we are in the laboratory, because you have to put so many ingredients when you make vaccines, because it should be nicely dissolvable, and it should be pH, right pH, and it should be stable, and it should be carry the mRNA couple of days in our body systems, and then it should be stable in the right temperature to our body system to. So many things. So when it comes to uh, the college students and those who want to explore the MD or PhD, so what direction we can move? So that is a couple of questions I have been here from the Mr. Cortelongo, so that we are going to see in the next session. um, can you uh, have, yes. So what are the biology areas we can explore when you go for the medical school or the college school? Means you definitely go for something nanotechnology based, because the nanotechnology is something really important, because that existing technology, macro technology, nanotechnology is something different. Why nanotechnology is all these vaccine molecules are the nanotechnology based. If you see that previous vaccines like Tdap, all these things, it is not in nanotechnology based. It is traditional molecules. Okay, that is the macro technology, I should say. So the nanotechnology is something very important, where you have to bring your science and technology stuff like that. And the biomedical technology, artificial intelligence, those things I would put in your plate to make this your education will be successful, because the education means we produce a lot of MDs and PhDs, all the peoples, they save lives, they help to survive. But not many MD, PhD doing something innovation. Okay, you have to be innovative where you have to put your science, your technology, your biology skills to solve some of the problems. For example, for simple problems, we have somebody has a hearing loss. We don't have any vaccines or drug to hearing loss. Okay, we should focus that what cause, what genes cause to hearing loss, what technology you can bring it, and what modification you can do in your genes, in your body system, in order to stop hearing loss. So the hearing loss so far, we are what the people are using is just electronics. They are using electronics and they amplify whatever you hear and they're able to hear it. But that is pretty much electro technology, electro electronics. But there is not in biology. Okay, so you have to put some biology in it. Okay, how the biology incorporate in your electronics or artificial intelligence that solves some of the problems, because the COVID is with the existing problem. We are going to have a lot of disease in in futures. So we have to identify that exactly what kind of virus going to come another 10 years, how to change your vaccines, and there is a billion of peoples, how billion of people's what are the genetic codes they have it, how the medical complication. So you have to know that all the data about that upcoming populations. Based on that, you have to design the vaccines, you have to design the drugs, exist emerging disease. So study something need to focus what is going to come in next 10 years or 100 years, what we have it, what technology we have it, what molecule we have it, how to take it. So those are the something important. So I would say that bioinformatics and nanotechnology and data science, artificial intelligence, machine learning, those are the something really important medical field that one should focus upcoming medical school or whatever. And second question is, what kind of careers do they lead to? Okay, so I would say that you should not stick with either physician or core scientist. You should be a physician scientist integrated with a lot of artificial intelligence and machine learning, stuff like that. Because machine learning, artificial intelligence gives kind of ideas exactly which molecule you pick to give that particular disease. Because a billion of molecules we have it, all these molecules, how we can put for particular diseases. For cancers, there is so many drugs are in the pipeline. So we should know that exactly what molecules would be helpful based on the data. So that is something you have to know, that AI and machine learning with the integration of the physician scientist background. So next is what are the popular college for the US? So popular colleges, there is, if you look at that survey, they will predict like a top 10 colleges for science, top 100 college for technology, top 100 college for the translation research, top 50 college for the cancer biology. So you should focus on this basis. So because each university, each college is specialized for some kind of topic. Okay, for MD Anderson Cancer Center, they treat with thousands of cancers. They have all the immunology, immunotherapy, all this traditional, all this treatment they have it. So if you want to go like that, you have to focus cancer centers, MD Anderson Cancer Center, or Memorial Sloan Cancer Center, like that. But if you want to do the something technology based, you have to think for Caltech, Harvard, Berkeley, things like that. Um, so how hard is to get into the college? The next question was how hard get into the college. So how hard means it is really hard getting into the college, current scenarios. So what I see, the lot of students applications in the college when they come to. So what we typically see is that how he is excelled in his academic performance. Pretty much all applicant has the academic excellence, but what extracurricular activities he has it apart from academic. So and then that we look at in extracurricular activities, what kind of internship they did and what he produced it. Is there any publication or he published something? Is there any innovation? Stuff like that we look at. So those are the things if you include in your resume, so it is easy to get in your college. That is the reason we have some kind of summer research. Whenever we get time, we do with great talented peoples. So as long as if you improve your careers with a lot of extracurricular activities, publishing it and making new technologies, bringing new technologies, and innovating something, that is the something easy you to get into the college. So next question is it important to get into the prestigious, highly selected college successful careers? Yes, that is something important because um, you have to have, because when we are selecting the college panels, we see that what innovation each applicant is have it. So we get into the innovative minds into the prestigious college. So so definitely it matters. So when it's going to the prestigious college, some percent it helpful to advance your careers, or getting good job and publishing your research and getting a research grant. So that plays some role, and I would say that definitely you have to focus getting into the some of the prestigious college, because if you see that some of the Nobel laureates, they from prestigious colleges, prestigious universities, working with the prestigious peoples. So that put you in the front ahead of other applicants. So definitely, I would look into the prestigious college when you do your research or when you pursue your translational research or MD or PhD, whatever you like it. So that's all about my presentation, and thank you very much for your patience and your time. It is really appreciated. So today I spoke definitely a very one layer of the molecules about the vaccines. I didn't dive in too much about this, how exactly purposed this vaccine, how it works, how it will be inside the spleen or whatever it will work. I didn't talk because this is something like public talk. It is not research talk. So at the end of the talk, I would definitely recommend some of the students to take a look at one of the book. My favorite book is the Molecule That Changed the World. That is the one of the really great book. You can learn that each drug in the polio or whatever the drug it came to right now, how much years they took, how many animals they sacrificed, and how many people are involved, because like polio vaccines, like it took 100 years. They sacrificed a billion of rats, a billion of pigs, billion of animals in order to prior to go to the clinical trials. So you should get kind of idea how this molecule works, what molecule they worked it, where they get the drug, which plant they got the drug, how they extracted the drug from the plants. So you kind of get kind of ideas all about molecules, drugs, everything. So I would definitely recommend all the aspiring medical students to take a look of the molecules that change the world. That is a wonderful book, because I teach a personalized medicine and the geriatric medicines. Before I take any assignment and any my teaching, I ask my students to read these books and come to my school and listen my lectures and do my research, because that gives the foundations to start to understand all the molecules. So that's all about my view about the vaccines molecules and my experience of reviewing a lot of vaccine documents of various pharma people's regulatory things. And I'm really happy to take any questions if you have it. Thank you so much for the opportunity to present my thoughts and my experience, which I had a little bit to hear. And thanks, Mr. Kotlengo and the Virtual Foundation and Lokesh and the Chokalingam and the rest of the participants. Thank you so much.
Thank you so much, Dr. Rajagopal. It's great to hear from you. I mean, I know it was very hard for you to reduce the content to our level and school kids level, and you tried your best. We can see that. It's like a lot of good information, and it's a great education for. I'm sure it's great education for all of us. Maybe we will go through. I see like some questions from the audience already, right? And also I have noted down some questions, from my end too. I will start with one of the audience question, right? Sarvesh has asked like, why is there a gap, dosage gap between first dose and the second dose? Pretty much it's there like in most of the vaccines, except very few, right? I think Johnson & Johnson, that's one of the vaccine came out like single dose, but most of the other vaccines come up with a 2-dose, and there are like specified time period. Can you brief us what's your
Yes. So that is interesting question. So why the dose difference? For example, today I take, just now I took vaccines. Okay. So what is this? The vaccine does it? It goes to our bloodstream. In bloodstream, we have neurons, hormones, nutrients, DNA, RNA, and so many thousands of molecules, thousands of components we have in the blood. So, and most important thing is we have the white blood cells. Okay, in our red blood, in our blood, we have the white blood cells. In white blood cells, we have the memory cells. Okay, so once you take these vaccines, it goes to your red blood cell in the bloodstream, and in the bloodstream you have the white blood cells. In the white blood cells, you have the memory cells. So you are teaching the memory cells this vaccine that to make antibodies. Okay, so if you take like 1 o'clock, you took your vaccines, like couple of like 3 o'clock, 4 o'clock. Okay, the memory cells start seeing your vaccines. So after 4 o'clock, the memory cells, okay, I got some mRNA vaccines. Okay, this vaccine, I'm going to send it into your body. Okay, I'm going to send it to the brain, spleen, pancreas, entire body system. I'm going to send it. Okay, and I'm going to process the rest of the cytoplasm nucleus, and I'm going to make my molecules. Okay, that is called antibodies. So it makes the memory cells need time, okay, to fight with any infections. So basically, why you want this time is you need this much time to teach the memory cells to identify the foreign invaders. So it starts some people, when I teach the personalized medicine, it starts some people, it starts like 4 hours right after immunization. Some people they get memory cells like 4 hours, some people get 1 hour, some people get 14 days, some people get 7 days. So that is the reason. Basically, your body, your immune system need to understand and map that whatever the foreign things, and then want to fight with anti bodies. So that is the reason we need this time. So once you get first dose, your memory cells, like 50% of memory cells will understand. When you give the second shot, pretty much above 90% of memory cell will understand that, oh, this is the bad guy, I'll start making antibodies. That is the reason.
Great, great. Thank you so much, Raj. I also noted down a question like, so you mentioned about the active and inactive ingredients, each of the vaccine and vaccine technology, right? And also how sometime it has like a, unexpected impact on the patients with underlying health conditions, right? So like some of them known, like, they have underlying health condition. is it possible to check with their medical provider based on the current vaccine set available, which will be a preferable vaccine for them? I hope the medical providers have knowledge about the ingredients used in the vaccine. Is that a possibility?
Yes, that is a good question. They definitely could check, okay, before I take any vaccine, I should check that what ingredients is there. And then is that suitable for me? Like I said, the potassium chloride, the people, for example, already there is a person who has a myocardial infarction because he has the high sodium or high potassium, all these things. But already this vaccine has also shown potassium chloride, everything, so that may be mess up the particular person who took vaccines. So those are the education definitely need the each personal or each patients before they taking vaccines. I'm sure pretty much some of the physicians have idea about what are the ingredients they have it. For example, Pfizer and BioNTech, they have like potassium chloride, all these things they have it. And if you see that Moderna, they don't have that potassium chloride. Definitely, the Moderna shoot for the people who doesn't have any hot medical history, all these things. So you have, it is not only this, and it has other lot of scenario also. So definitely one should check with before taking whatever the vaccine. That is what you take whenever you go to the doctors. They say that drink a lot of water. Drink. That means if you take a lot of fluids, if you have some kind of potassium chloride, all these molecules, it dilute and doesn't give any effects. So that is the reason intake of fluid definitely helps. Based on this, all these data which we reviewed before the vaccine come to the market, I think it met all the safety standards, and it met all the parameter which is set to the FDA or the rest of the physician committee and the safety committee profiles. So there is no issue. Pretty much all vaccines are pretty much safe and doesn't give much effect. But the people who has medical condition and immune issues, those people, it gives a lot of issues. Pretty much, I think they should check before taking any vaccines. There is no issue at all.
Okay. And I see one more question come up on the chat, like why does vaccine need to be stored at such low temperatures? That's from Jyotsna.
The thing is, we don't have the technology, okay? We don't have any technology to keep the vaccine low temperature. That is the reason they added the PEG molecules under fructose, the sucrose molecules under phosphocholine. Those molecules helps to keep the like 7 degrees Celsius something or minus 1 degree, 2 degrees. So we don't have the technology to keep the low temperature. That's how the participants and the people, young minds, need to think about that. What technology we can bring the lowering the temperature for keeping like years, stuff like that. Whatever the technology we have, like a couple of weeks, 1 weeks, I think so we can keep, or 2 weeks stability. So we have to advance the technology to keep the vaccines low temperature for at least 2-3 months, so that it can be able to shift one place to another place. And especially a couple of months, I had a talk with one of the defense scientists that they want to take this vaccine to the warfield soldiers, how we can take and what are the technology we can improve, what are the things we can do. So there is a lot of talks are going on about this. So definitely we need the technology, how we can purpose the container that can hold for a year this technology under low temperature. So definitely lack of technology. We don't have any technology.
Okay, I also see a response from Mr. R.G. Singham for that question. Thank you, Mr. Singham. So anybody have a question? I see one more question is from Prerna. Please put it in the chat. we don't have much time. Like we have already crossed 11 o'clock threshold. So maybe we can go through like another 2-3 question, and then we will have to wrap, right? So I see a question from Mrna. Is there a difference between the COVID vaccination given to different age cohorts, such as people 12 to 15, and there are like always adults, right, more than 18? Are there any difference in the vaccine? That's a basic question.
Yes, whatever the vaccine which we are invented, that is the common vaccine. It is not a pediatric vaccine. It's not a geriatric vaccine. So we have to find the geriatric vaccines for the COVID and a pediatric vaccine for the COVID. So there is a big things going on the pediatric vaccines, and then some of the Johnson and Johnson and the Pfizer companies, they are working with small newborn mice. That is for the pediatric mice things. That is the old-aged mice or old-aged mice. So there is a different kind of vaccine works under the clinical trials. Hopefully, another couple of months, it will be able to come to the market, dedicate for the pediatric COVID vaccine and dedicate for the geriatric COVID vaccine. So still under the clinical trials.
Okay, great. Thanks, Raj. And moving on to the career side, you talked about the career options for biologists. So you worked on the vaccine development team, and you've been part of it, all of that. What's a typical educational level needed to be part of these vaccine research and development? And as you mentioned, you already clearly mentioned about it's not just biologists, right? There are like a lot of other like professionals also needed to be part of vaccine development, research and development. So if somebody have an aspiration to become or contribute to this kind of program, what's the like approximate education level, like a undergrad or a master's or a PhD, and what kind of field, right? Is pure biology helps, or biology with any other supplemental technology related or a science helps in that area?
Yes. To keep this question short, say this virus, whatever the virus, it is pretty much coming from the environment. To treat that diseases, the drug and the vaccine also coming from the same nature. Okay, so we have all this in the nature. Okay, but where it is there exactly, we have to find it. Okay, like for headache, if you get a headache or fever, we are giving Tylenol. Okay, Tylenol is chemically modified, but the source is somewhere in the nature. Okay, so to develop the Tylenol, this medication, there is 100 people they work, like in the history. They don't have like a PhD, or they don't even finish, some of them with a undergraduate in the past. Okay, at the time they don't have nothing. They had only interest, and there is no technology, but still they are able to success and solve the problems to treat a variety of diseases based on the experimentals, all these things like preclinical, clinical, all these things. But now the trend is changing, and each one should know a lot of things to talk. Okay, right now, if someone want to become a vaccine scientist, okay, there is a lot of criteria there, and you have to pass. For example, I want to apply NIH grant for the SARS-CoV emergency grant to test my animals, okay, my vaccines in here, my laboratory. I should follow a lot of protocols. How I am going to give the vaccines, what mice, what clinical trials I'm going to take, and what are the protocols, how much I'm going to take. All these things I have to document it, and I have to submit to the regulatory board, IRB, stuff like that. So yeah, become a scientist, you definitely need the passion and the interest. That only very important, but that only not enough. you should know that how this, because science and technology we can put together. So I would suggest something like be like a physician and scientist, okay, with the information technology, data science, artificial intelligence, bioinformatics. Whenever you get whenever get time, you try to learn whatever possible, as much as possible, about science and technology. It is hard to study and hard to learn everything, but you need to have the interest to learn everything. Then only you can solve the problems. For example, this COVID, this is the team, big team, okay, the big team played to solve the problem, okay, data scientist, bioinformatician, vaccinologist, artificial intelligence, like basic scientists, like so many people played in this role. That's how we could able to get this vaccine very fast. uh, Same thing, we should know that pretty much try to learn as much as possible and put your interest what you want to solve, and talk with peoples and follow the literature. These are the essential criteria. If you follow up to date every day, you definitely will be in big shape, and you can big noble or it. So basically what I'm trying to see is the interest is very important what you want to work.
Great, great. Thank you, Doctor. I see couple of more questions on the chat. With that, probably we will wrap it. I know it's really running, more than what we expected, the time threshold. Mr. Chokalingam asked this question: do you believe there will be a pathway to do animal testing, free vaccine, and medicine development? Is there, without animal testing, a vaccine development, is it a possibility? Is there a pathway in progress?
No, no, without animal testing there is no possibility, okay, 100% I could say that without animal testing we cannot able to come to the clinical trials, because when we want to see the clinical trials it should be bossed. There is a 4 levels in animal trials: BSL1, BSL2, BSL3, BSL4. It should pass all the 4 levels to come to the clinical trial levels. So without preclinical we cannot take into the clinical trials to perform any vaccine development or medicinal development. So people, that is to cut that preclinicals, we need technology, okay. Based on the data, we have the billion of data in the literature. So all the data, make it is like a AI or data science based model, and compile and put it in the like big data. From there bring the molecules in order to cut that preclinical trials. But that is still it's hard, because previously, before the COVID, when it came, we talked with a lot of AI guys and they gave some molecules, some drugs, okay. We synthesized those molecules, those vaccines, we put into the animals or cell culture, but it doesn't give any supportive evidence based on the AI or machine learning. So it definitely got failure. But we worked with a previous literature published value molecules, and it worked a little bit. So we cannot ignore the technology also, and we should not ignore the preclinical. So it should go both hands on hands.
Great, great. Thank you. Thanks for your answer. And this could be the last question for the day from Kalpana Tirumalai. What are all the off-life of different COVID vaccines?
Yes, this one is the based on the Pfizer data which we reviewed. It is like a couple of days. It will be there in our body biological systems, and whereas because we want to the basically this mRNA vaccine, whatever the vaccine is, we have to train our bodies. Okay, after training, it should come out from the body system. That is the purpose. It should not stay forever in our body. Okay, that is not purpose. If it is stay, it gives another impact, or it gives another problems in our body. So pretty much it is couple of days it stays in our body. Pretty all vaccines.
Great, great. And, I think some people want to contact you offline. Is it okay in case if, Kotelanga or Chocolingam choose to share your contact, you can let them know offline. We can communicate that. And a final note, we also, me and Kotalango come to know about you do some programs for the high school students, right? Do you want to give like a brief, intro about that program, Dr. Raj?
Yeah, actually, I have been working with a lot of MD and PhD students over a period of years. And I thought of why don't we talk to the young mind also? It will be give additional impact and to solve some of the problems which we are going to face in another 100 years or so. So I want to talk pretty much young mind also, because these MD students, all they are busy with a lot of rotations and patients, residential, all these things. So I want to change something. That is the reason I thought of I will talk to some of the young students in order to boost and make them to understand. So they can contact me. My email ID probably, or they have my LinkedIn or Facebook or whatever. They can reach to me, the particular students. This I recently started about this high school, the summer research. So that we can do it. The interested people definitely can approach. And if the right candidate, they'll get opportunity to in order to show their potential to the research community, how caliber about their critical thinking and their technology, their all these things we can able to focus as a publication. They are welcome to approach either LinkedIn, or you are welcome to share my email ID, the person who are interested. I'm okay, I'm good with that.
Great. Thank you for, for willing to contact and work with the younger generation. That's a very welcome note, and, great message for all the students who are eagerly attending this session. And thank you so much, Dr. Raj, for patiently answering all the questions, and, even to take it further, post this meeting session, right? It's great message from you. Kotalango, you want to add anything to it to conclude the session?
No, yeah, that's great. I thank you once again, Dr. Raj, for your patience and time in talking to our audience here. I definitely learned a lot. Now, when you said vaccine for hearing loss, I'm like, I only heard vaccines for like flu shots and diseases like that. The future is like really interesting, right? Where it's all about protecting before things happen and what other things can be invented in the future. And there's obviously a lot of scope for young people who want to enter this field. So that's awesome to hear. And once again, thank you everyone for joining today. Thank you, Raj, for information. Thank you, Logesh, for moderating the conversation. And Dr. Chocolingam for hosting the webinar. Thank you all.
Thank you so much for thank you so much for the opportunity to interact with the young minds, and it is a great to meet you all, and thank you so much for the opportunity. Appreciate it.
Excellent.
Thank you.
Thank you.
Thank you all.
Thank you.